Good morning, Amos.
Good morning.
Good morning, Yehuda.
So, may I suggest that first you briefly introduce yourselves, and then we'll embark with our discussion. So, Noah, please say a few words about yourself.
Okay, hi. Ken.
You are the younger, so more important than that.
So I'm Noa. I'm an illustrator and a graphic designer. I live in Haifa. That's the most important. No, I have a 1-year-old, Alma. I'm the daughter of Amos.
And Amos, who are you?
I'm father of Noah, you see, and I hope you agree with me that she's so charming, no? And she is the artist of the family, also her mother, also the late grandfather. Only I am the only one that is ignorant regarding art, but I hope that she will succeed and she will make nice career by painting or who knows what else. So I'm a doctor, a physician, MD, a dermatologist that also focus all his time and devoted a lot to research, mostly basic research in dermatology and in immunodermatology. And I continue working many years after the retirement in my clinic, public clinic, not private one, and also as an investigator. I'm running now a small clinic in Rambam Hospital, and also in the Technion, I'm running a laboratory. So I share my time on clinic on one end, and on the other hand, on research. And that's all. What can I say about myself?
Well, I may add just a small detail that you are also a former Director General of a large geriatric hospital.
Yeah, that's true.
Ran for many years.
Yeah.
This is part of the assembly that we are trying to show today.
Yeah, but the life is so, and people that after retirement will tell you, because I know that you also, but you retired only a few months ago. But you will see, I have retired, I think, officially, it's 10 years now, exactly 10 years. So, and you will forget. I have forgotten the hospital and running hospital and being director of hospital. The life, it brings you new things, and it's a new area for me. So, so I have forgotten the time being in the hospital. I have not forgotten the message that, because it was not a simple geriatric hospital, it was a rehabilitation one. And the message that it's important to support aged people, old people, should be running all the period for the human being. And that's what's important. That's what was important for me when I was running the hospital.
Okay, so perhaps we'll begin, Amos, by giving us an example of your work. I know that you are international renowned researcher on skin research, especially immunodermatology, and one of the things that is known about you is the innovative models that you created with your staff at the laboratory. So perhaps we'll start by seeing an example of such a work.
Yeah, that's a good idea. Established laboratory many years ago, almost 40 years ago, and the laboratory is located at the Technion, Haifa. And my hobby is to develop models for various diseases, skin diseases. And very popular one is aging one. And I would like to focus on this. Can you show the first slide? So now this is a list of diseases that that my team and I have succeeded to develop or to establish during the last 3 decades. You can see here. And as I said, I would like to focus on the aging one. So first of all, in order to, the models are based on humanized one. And why humanized? Because we will see in later on that the mice, they are different from human. Most of the studies all over the world, the preclinical studies, are based on mice. But there is a big difference between mice and human. And therefore, I decided, as I said many years ago, to develop humanized one, in which I transfer elements of human into the mice. But in order that the mice will exist, they need a specific pathogenic-free room, so a very sterile room. And you can see myself with a member of my team in the rooming in this pathogen-free room in which the mice are located. Very special mice, very expensive, by the way. And we have to also, as we can see, to wear protective clothes. Okay. Now, regarding the aged skin and the young skin, I will speak very clear, and I would like to make it very superficially so everyone can understand what I say, that if we look under the microscope on human skin, we can see the histology specimen under microscopy. We can see it's like a desert. You can see that the aged skin lacks most of the cells, most of the blast cells, most of the elements that we can see in young skin. Less blood vessels, less fat, as we can see it here, written subcutaneous fat. So all this is very deserted and very thin. And therefore, indeed, when we look or touch human aged skin, it's very touchy, it's very thin, it's very sensitive. So the question is, can the skin become younger? I know it's a very popular question, but the question here is not in a clinical issue, but mostly on the basic one. If histologically or molecularly, we can change the behavior of the aged skin. And in order to address this issue, we obtained biopsies from old people and also of young as a control, and transplanted it onto young mice. As I said, very special young mice. Each mouse lacks all the immune system. So this mouse, the special mouse, has a potential ability to accept the donor from the old individual, from the old donor, yes. So we obtain, as I said, samples from old person and grafted it or transplanted it onto a SCID mouse. And what did we see? Yes. First of all, we can see macroscopically only. No, you make it too. You can look and you can see immediately, without taking out or removing the skin samples from the mice or from the mouse, you can see the changes of the aged skin. It becomes younger, less wrinkles in the aged samples. When we grafted the old skin from the old person into old mouse, we can see that the skin hasn't been changed, the same pattern, the same wrinkles. While when we transplanted the old skin from old person onto young mouse, we can see very huge differences between both skin, both samples, on the old versus the young mice. And we look at special staining of the specimen, of the histological specimen, which were obtained from the samples that were on the mice. You can see before transplantation and after 1 month later on. Only by looking at the staining, you can see the difference, the big difference between the two samples, the two examples. And what did we see? What we see here is the rejuvenation process, some rejuvenation process, because each staining present the energy, the energy of the cells within the grafted tissues. So, okay, I can also to give some examples. PGC1 alpha, per example, this one controls how cells generate energy. We can see or if we can above a bit, PGC1 alpha I mentioned just now, MTCO1, the last one, is essential for energy production. It controls the energy within cells of each tissue, in other words, of the skin. So I have to make it very, I would like to make it very general, but we can see also here the striking difference between aged skin grafted onto young mouse, as you can see in the right of the picture, versus the before engrafted or versus aged skin on old mice. You can see the big differences regarding the staining. For example, this one, the last one, is a collagen, which is a huge protein, a huge importance to give the strength and the ability to stay and to survive to the skin.
So about the collagen, have you also looked at the arrangement of the collagen fibers? Have you looked at the three-dimensional structure of the skin, or just the one?
No. That's a good question, and we show it very in the right area. You can see the figures. You can see it's written thin filaments and thick filaments, and you can see the huge difference between old skin in old mouse versus old skin in young mouse. So yeah, exactly. When we transplanted the skin, the aged skin, the old skin onto young mice, the pattern, the morphology of the collagen becomes exactly like as we see in young skin. And here also, no, that's nice. You should look very carefully. SIRT1 and MTCO1, which play very important role within the cells of each part of the skin, and both play a role in the producing energy for example, here, what we see is the upper part, the staining part that we see here, one is a blue, and thereafter it's brown, something like brown, and thereafter again blue. This is called the epidermis, which is the upper part, the upper layers of the skin, composed of cells named keratinocytes. And the third one, and MTCO1, is essential. The third one protects cells from aging, whereas the MTCO is essential for producing energy in the outer cells, in the epidermis. So we can see very clearly that in old skin, in aged skin, there is down-regulation, decreased expression of these markers in the epidermis, in the upper layers or upper part of the skin, indicating that the skin possesses less energy. But when we transplanted the skin onto young mouse, as we can see it here, you can see the brown staining, indicating that there is upregulation, there is covering or recovering of the aged keratinocytes of the upper part of the skin. So the upper part of the skin following the transplantation has better health, if we can say it in popular words, because they had a lot of energy, in contrast to the skin before transplantation, or in contrast to aged skin. But very interestingly, because when the mice, the aged mice, we maintain them for 1 year after transplantation. So even the mice, the young mice, they become older and old, because 1 year for mice, they are old. And we can see that the aged skin, that after 1 month become younger, the skin become old again after 1 year grafted on young mouse that become old mouse. So the life is here. In any case, it will be changed. Even if we make younger the skin, thereafter it can't be forever.
Okay.
That's the message of this staining. Oh, okay. And here we can see what we call in science the quantitative data, showing the upregulation, the increased expression of the activity of SIRT1 and MTCO after 1 month, and the decreasing decline after 1 year, being on the mice. Okay, so now this is a critical observation, because if we look on the aged skin on young mice, here it's mouse, not mice, because it's one. So aged skin grafted on young mouse, we see changes, very critical changes of the morphology of the staining as we observed just now. But this is a special staining. It's a triple staining in which we used several markers in the same staining. And what do we see? We see a significant increased expression of the blood vessels in the aged skin on the young mouse. Because one of changes characteristic to old skin, but it's not only old skin. Unfortunately, other critical organs in the old person, we can see the decreased flow up of the blood within the blood vessels, and we can see decreased number of blood vessels. Here in the skin, we can see the differences when we look at our right. When we looked, and we can see the scarcity, the few numbers, if at all, of blood vessels in aged skin on old mouse. So striking difference. So this transplantation on young mouse or young mouse versus old mouse illustrate to us very clearly the differences between the blood flow and blood vessels in aged skin versus young skin, and the ability of the blood vessels to recover following the transplantation. So it's important for us to know that there is a potential ability to the blood vessels to repopulate, to recover, even after being in aged skin, and after being depressed for many years, relatively. And after 1 month, we can see so significant and striking difference due to very simple procedure. So I know how to do it. So this is another staining, shows a huge difference. And we can see here the quantitative data showing what we showing very clearly the difference in numbers. And the questions that we asked ourselves, how can we explain this repopulation or recovery of blood vessels following the transplantation? And the answer is that there is a protein known as VEGF, vascular endothelial growth factor, which is a protein that stimulates the growth of new blood vessels, which is crucial for supplying oxygen and so on, especially during wound healing and etc., and blood flow, as I mentioned just now. So the question is whether VEGF, this protein, this very special protein, plays a role in all this process, in the recovery of the aged skin following the transplantation. So we made a special staining, also here, triple staining, white triple, because we used 3 markers staining on each sample, skin samples, under the and thereafter we looked under the microscope, and we used cells. We perform specific staining or using specific markers to stain the cells that are known to produce this VEGF, this specific protein. And indeed, we can see that all these cells, important cells, located within the dermis or epidermis of the skin, we can see upregulation, increased expression of this protein, VEGF, by keratinocytes that I said just ago, that they are the main cells that make up the outer layer of the skin, providing protection and helping to keep the skin strong and waterproof. The macrophages, which are clean the harmful invaders, and the platelets, which are very important cells, not only in the skin, but in other critical organs, mostly in the blood, which helps stop bleeding and heal woundings, because each one of these cells possesses the ability to produce large amounts of hormones, of proteins, and etc. So we can see that these cells, after transplantation onto the old or to the aged mice, produce large amounts of this VEGF. The quantitative data showed very clearly the differences between graft before transplantation versus grafting the aged skin on old mouse or old mice. And here we can see the increased production of this special VEGF when we transplant the old skin onto young mice. So we assume the VGF, now not only assume, we know that VGF significantly upregulated following the transplantation. And here we can see again the expression, the huge expression of VGF by the keratinocytes. And the keratinocytes, it's several layers. As I said, it's the upper part of the skin, very compact areas. And they produce large amounts of this protein, which play crucial role in inducing proliferating more and more blood vessels. I mean, induced or increase or improve the blood supply by inducing more and more proliferating blood vessels. So now we perform another mode of experiment or mode of staining, because I will not speak about it, only say that we detected the expression of RNA, which is within a part or component within the nucleus of each cells in the dermis. And we found out that the upregulation of VGF is also linked to upregulation or increased production of VGF by the mouse skin. It's very difficult here to see it, but you should believe me. So what is the conclusion of this experiment? Because of the transplantation, the human skin stimulate to increase the production of VGF. But who stimulate this production? Well, the stimulator here is the murine, the mouse VGF, the mouse, the young mouse that produce VGF, murine VGF-A, and this normally produced because it's a young mouse. And this young mouse that produce VGF penetrate to the human skin, engaged, bind to the VGF receptor, the human receptor, the human part of the VGF, which is located in the membrane of cells in the skin. And this stimulation leads to production of more and more VGF, as we can see it here. So mouse VGF stimulate human VGF receptor, or receptor is a kind, that it's enable the cells, the human cells, to bind to the mouse VGF. And this combination, this engagement brings to positive feedbacks. In other words, stimulate the skin, the cells in the skin or the tissue in the skin to produce more and more VGF. This engagement has been described previously. It's not our findings. In any case, VGF do not induce proliferation, do not only produce proliferation of blood vessels, but also stimulate many other cells to become younger. And here we can see that there are other parts that becomes stimulated because of the mouse skin, the mouse skin possess ability to stimulate other parts, not only the VGF, but we'll ignore it today, now at least. So
I just throw a quick question about the stimulation, the potential stimulation, that VGF may also stimulate malignant.
Yeah, I will come it. I have a slide on it. That's true. This is a problem. This is a problem because it's prevent us to use VGF in a clinical trial, or in any case. That's a good, yeah. But in any case, what in very short or brief sentence, we can say that this model the skin recover due to, and the first step is due to the murine VEGF, which initiated all the rejuvenation process of the aged skin following transplantation onto mice. And this leads just what you mentioned just now. This leads us to the question about the clinical relevance of all this experiment. Can we use VEGF in people? So the answer is no, because just as Yuda mentioned, because of the neoplastic proliferation and invasion. So the problem is that VEGF encourage, stimulate malignant tissue to repopulate, to regrowth, to growth, and therefore it's forbidden to use it. And we have to be very carefully by using this VEGF. But not only regarding tumor, but also it leads to, it induced skin phototoxicity. It can lead to psoriasis, exacerbation or flare-up of psoriasis, because I don't know if not only one knows on aware about psoriasis, but VEGF play a critical role in the development of psoriasis. And therefore, treatment with VEGF can lead to flare-up or also develop psoriasis with people with appropriate background. And also, it's associated with senile cataract. And also, and this is also quite frequent, it has been observed several times, at least in animal models, that VEGF can induce autoimmune diseases. So we have a lot of risk to use it, many side effects, many potential side effects, and therefore we can't use it in a clinic trials or in clinics, in general clinics. So how can we translate these findings into practical anti-aging medicine? We have to find a topical, as it's written here, some VEGF, like VEGF, with minimal systemic absorption, and to use it in people that are not, don't have any background of psoriasis, or have no any risk to develop tumors. We can use some VEGF-like treatment for short-term period of time. But nowadays, we are now working on developing, but because short time after finishing this experiment, this study, and after publishing, we found, my team and I, we found molecules that stimulate the markers that are usually stimulated by VEGF, but can be also stimulated by some compound that is very similar to VEGF, but it's not VEGF. So now we are conducting a preclinical trial, and I hope it will work out. And but we are not the only one showing the potential ability of VEGF to the rejuvenation. Very known expert, very known scientist from Jerusalem named Professor Eli Keshet, but he published in Science 3 years ago. He used a very unique mice, and he treated them by VEGF and showed a complete rejuvenation of these mice. But he didn't mention the skin. So we were the first to show the skin effect by VEGF. But he showed all the critical organs. The rejuvenation of VEGF in these special mice, rejuvenation of all this, or improved improved activity, or no, better say, improved function of all these critical organs in aged mice following activity of VEGF. And so I have to quote a very nice sentence that aging was once thought to be unavoidable, but now it's increasingly believed that, I can't see what's written here because technical problem. So aging was once thought to be unavoidable, but now it's increasingly believed that treatments to delay it are possible. I will call it again. I will say it again.
No, it was very clear. It was very clear, Amos.
Okay, okay.
We can see it on the slide.
Okay. So now all my colleagues, Dr. Aviad Keren, and he's the main investigator in my laboratory, Professor Dr. Yuda Ullman, and Ralph Paus, also not mentioned here because his name is here, but picture, I don't know where is it. Very known in science all over the world. So that's all my colleagues that helping me a lot in developing all the models and also this last one. Thank you, Yuda.
Great. Okay, so let's just, I reclaim the hosting. Yeah. Okay. Amos, are there any models in similar models in other animals? Suppose we take an animal that is known to be able to rejuvenate the skin, like
no, I can. I have slides here if you want me.
No, just you know, a short answer.
Yeah, there are several, not only mouse. There are several mouse models for rejuvenation, but each one of them do not cover all the aging aspects, have a lot of defects. I can give you one example, because if you know, for sure you are a big professor. We have not to, again, we have to mention it. So the telomere of the of the DNA, in which also, it becomes shorter and shorter according and related to the aging of the organ in human. Yeah, it becomes shorter and shorter. But for example, in mice, it's the opposite. And telomere is one of the main target issue for detecting aging process. So if we know that in mice, it's different, so striking difference between telomere activity versus human, it's a good example. It's an example to show why we can't use always mice as a model for aging, for investigating aging. And therefore, it's so important to use humanized mice.
Okay. Noa, are you aware of those studies in such details? Have you been exposed to those that information during the years?
Yeah, most of them, yes. Sometimes there were a few times that my father practiced on me, right? You told me, showed me his lectures, and he performed it to me, and I found it very interesting. Not every subject, but this specific one, yes. I've heard it before. It's very interesting. And also, I think once I made, when I was in high school, I made a big, I had a big project about one of his research about alopecia areata. And
Yeah.
I think I understood it. And,
Yeah.
Yeah. Also, when I was younger, I was at your lab. and I saw the mouse. Yeah.
Okay, so I know that one of your you are very innovative and well-known illustrator and designer.
Thank you.
But I know that one of your projects is involved with retirement. So perhaps we can discuss a bit that project, if you can show us, and then we'll try to combine everything together.
Okay, yeah.
Okay?
Should I open the pictures now?
Yes, yes, please. Let's see it.
Okay. So
Yeah.
yeah, I made a project. I was influenced by my father's retirement from FEMA in the hospital. And for a long time, I saw how he, like even before the retirement, like a year before even, I could see how it affected on him and how it influenced him, and how every day was just, he was expecting the unknown. Now, retrospectively, I can say that he's working more and he's more fulfilled, and I don't think it had any negative influence on you. No, I think so. But
only on the beginning.
yeah, but back then it was like what I'm going to do, and like I could see that a person comes to some point where like you tell him, okay, you need to stop doing what you did now. You had enough, you finished. And the age is not in the same, it's not like how you feel. Well, I don't express myself so good like you.
No, it's fine.
No, it was clear.
No, very clear. Make it very good.
So I interviewed some people that retired or at a certain age, or were close to pension or went to pension, and then I just I wanted to represent a few ways to deal with this period of time. So yeah. Like I think most of my illustrations describe maybe something like from your inner self, maybe loneliness, or maybe, but because I think it's something that you deal with yourself. It's like questioning yourself about your life, about what you did so far, and what are you going to do. I think you have to deal with it. Some people deal with it in a positive way, like, but for some it was more lonely, I think, or maybe searching yourself, searching a new activity, how to spend your time if not working. So maybe take some classes, maybe doing some sport.
This one,
yeah,
is very similar to me, no?
I think I used you as a model.
Yeah, that's I want. Yeah. When I look now, I can see
Yeah,
myself.
but I changed you a little. It's not you, but I used you as a model,
I think,
as a reference.
No, I think
Yeah,
frequently, sincerely,
yeah,
that I have a bit more hair, no?
yeah.
So
Absolutely.
why so?
Because it's not really
It's
you,
not
but
myself, yeah.
I needed
But I look this one also, but I don't know to dance, I'm
I
someone that don't dance. But here again, so few hair.
No, it's not you.
It's like my father.
I know.
Yeah, okay.
So now when I'm looking at those pictures that I did, I see a lot of emotion and loneliness, and I don't know, something very touching. So, and I think this is what I want to represent, that to deal with this issue in, I don't know, in some, with compassion, and yeah, like I wanted to show like some are investing their time in their grandchildren, and some can't sleep at night overthinking what am I going to do. I think this illustration was deeply connected to my father as a person that I knew that, you know, his mind is fully on this matter, right?
Yeah, but this is it's not me.
No, it's not you, but I mean like the main issue here.
Yeah.
It's not you.
But you have never seen me or my
But I knew.
Or with my wife or your mother together like this. Never. Yeah. And this one like look like completely desperate and very sad, and I have never seen myself like this.
But I think I took like some idea and I tried to represent it, maybe even more extreme. But to make a point, sometimes you need to go a little more.
Okay.
You know what I mean?
Yeah, I know. I understand you.
And or someone will go and travel the world and experience more, and it's like you know, some people that leave everything, and
I'm clumsy if I'm standing this point to me.
I know. And the idea was at the end, because I made a book, so the end was everyone together in the pool, and yeah, basically I think this sums up everything.
Yeah, but this one also remind myself.
Yeah.
Only he's also again without hair, complete baldness, and very fat.
It's not you.
I know.
No.
But when you stand this one, you thought about myself when you doing.
Yeah.
You thought about your father.
Yeah, but it's not you, but yeah.
Okay.
And well, what I wanted to say. So I dealt a lot with something more, how do you say, a state of mind of the soul. And well, and this is not from this project, but I wanted to show more things, like, again, the loneliness, the you know, the something unreal that is only like something that you experience. Yeah. Okay.
Yes.
Yeah.
Great.
There's something more that I can say.
Yeah, but this is like the darkness of the life, no?
Yeah. Yeah, that's what I'm trying to say. Like, it's more.
When I looked at it, it's the darkness of the life.
It's not only darkness,
no?
Because people are dancing, but I think it's everything. It's also hard and a struggle, but also together and also alone. And
yeah, and this one.
Yeah.
Also alone and desperate.
But maybe they will meet together.
You think so? Okay.
But I think a lot of times I deal with things that are, it's like unreal, because it's something in your mind, a state of mind.
And so.
In your soul. Yeah.
Okay.
Yeah. So I'll go back to you.
Okay, so are there more images now?
No.
Okay, so I just turn to this frame.
Mine was shorter than yours.
You know, it is interesting to see that most of the figures in images that you chose to display are with a lot of hair, despite their aging, but the hair looks much younger. And Amos, you immediately responded to it, because I know that you are also very much into hair research, which we didn't address and we will not address this time. Was there, Noah, any particular message or emphasis about the hair, you think, in your work?
I don't think I thought about it too much. And also I got some reviews. Someone told me that the way I painted it with these little circles, so they told me it's like a hair also, you know. So, but no, I don't think I thought about it too much. I think I made one or two men without hair, and yeah, I don't think I talked about it so much. Yeah.
Another question is about your work in general. Do you apply much attention to the figure's skin,
Yeah.
or is it a major issue you try to address more attention to the skin?
I try to show variety if I paint people, like a lot of people, or even a few people. Yeah, I think about it. It's like I do pay attention to it, to the skin color, to show variety. Also, I think now I do think more about the hair, like to show, like sometimes I do people without hair. Like I feel like now I think about it more in what I show and represent.
I noticed looking at several of your works that while on the skin there may be a lot of freckles, but not much wrinkles. Is it intentional?
What in the journal things? I don't know. Maybe I haven't thought about it. I don't think they see so much details in the works that I do in the journal. People usually the figures are small, and yeah, I don't go to that much details, I think. Well, in the journal, no, because you can't see it so much. Yeah.
Almost when
Maybe.
yes, sorry.
No,
Please.
I remember I did one cover that we had many characters, and then I did like noticed if I'm making someone more older, so we had more wrinkles, and then it was really specific. Yeah.
Okay. Almost when you look at Noah's works, what do you think? I don't mean it's wonderful. You told me a lot about her when we first met, but as a scientist and as well as a father, what do you think about her works? A series of works about retirement. What do you feel?
Well, I told you in the beginning that I don't understand. I'm not an expert, and I don't understand from, my wife. She is the artist, and she likes to go to museums and everything. I'm going with her, but when I'm looking at pictures and arts in the museums, I don't understand nothing. It's for me, it's nothing. So. But I like her, all what she did and what she does and what she will do. And that's make me happy. And I want her to make a career. That's what I can say. But to give you an answer, a good answer, it will be not correct. It will. I don't understand. So
Noah,
I'm
do you
sincere
think?
with you.
Okay. Noah, do you think that your father doesn't understand anything about art?
No, I don't think so. I think he understands, but sometimes
Wow, that's true. She asked me a lot.
because
And she sent me always through the WhatsApp. Before she didn't say, but she's working in Globes, yeah. Once a month, she sends them illustration.
Major financial newspaper here.
Exactly. And so has never sent them before sending it to me and asking my opinion. So
I think
so
no,
and I answered always.
because
That's true.
I think he has a good opinion, and sometimes I think even someone that is not from my industry can give me can guide me even better than someone that is in my industry. And I think even in school I used to send you my pictures.
Yeah, that's true. Always.
Yeah, so I don't agree. I think he has a good eye, and like in the important things, like to tell me if it's good or not. And
But tell you what I wanted in the beginning.
what, for me to be a doctor?
Yeah.
Yeah.
Yeah, he will be interested because he has his own children.
Yeah, I
Tell him.
Why? You
Yeah,
just did. Yeah, I think it's your greatest disappointment.
No, I have overcome it.
No, I'm
Yeah.
kidding. Yeah, but I'm really happy.
I wanted her to become MD, a physician, and I begged her to become to go to school, to medical school, and she
I was in a conflict.
she was, yeah.
But
Okay.
no, I'm glad now we can collaborate.
I have a question now. Our project is about art and science, and we are looking into the possible collaborations between artists and scientists. Now, suppose, Amos, you are able to accept as a co-worker an artist to your laboratory. You could you can host an artist or a few artists in your laboratory on a daily basis, not just as a visitor. Do you think it would be helpful for you? Because your models and your work is so creative. Do you think you need other creative people from other disciplines? Would it be
Creative people? It's important. I think not only for me, but for many laboratories. I think it's a nice idea. I'm not sure that maybe a creative science that's important. I'm not sure about about it. And theoretically, if someone come to the lab, not important that it's my lab, it could be of another one. What will he do there? What will he do? Because it's a Technion. We are working in the lab. It's all the experiments are all, when I show it, it become very theoretically and very nice and very even nice pictures to look at it, nice staining, but practically when someone like this comes to the lab, has nothing to do there. I think so.
okay? And Noah, do you think that in your studio, would you think that having someone with scientific background or scientist coming regularly to work with you, think with you, discuss your ideas, do you think it would be helpful for you?
Well, maybe sometimes, yes. I think that a lot of time a scientist must have some like a creative way of thinking. Though I have my father as an example, and he helps me a lot. And I don't know if, like, also in my field, it's very technical, and you need to know what to do. But maybe as an advisor or something, maybe possibly.
But I have something to comment about your question. When you gather together all the data, and you are in a science and you are a physician, and So you understand what I say now. When you gather it all together, and you make, you want to make a paper, you may, you know, to publish the data. Nowadays, it become more and more popular, and you know, the very prestigious journals, they are asking to make figures and illustrations. And now we can tell you, I make illustrations alone, and despite of all this, I'm a clumsy person, unfortunately, but I make a lot of an effort to improve myself, to do it alone. But, and I don't want to bother, she's so busy with her dog. Yeah, sometimes, but all the time I'm making it alone. But it's, you know, if someone with a creative person can help us a lot, you know, for gathering the data, for making it, for making a nice illustration for the publication, that's a good idea. And even I thought about it myself several times.
Okay, guys, I think we had a very good session, and I think we learned together a lot. And thank you very much.